We are glad to have you here. I'm working from home, so this is my puppy, Percy. You might hear him a little bit, but I'll try to mute myself. Discoveries in the Making is an event that the graduate school puts on. It's a special project created by Dean McMahon to show off and highlight our graduate students' research, particularly to allow them to have a chance to present it to a lay audience or a general audience. This program is the definite special pet of Dean McMahon. We have been going strong through Zoom, through quarantine. We've had tons of people join us that would have never been able to, so definitely a silver lining, a small silver lining of the pandemic. So I'm Dr. Sarah Creel. I am the senior writing instructor within the graduate school, and I've had the pleasure of working with these students and postdoc from the CURE grant. I'd like to introduce you to Dr. Jennifer Pollock, and she's going to say a couple of words about the grant, and then we'll get started.
Hi, everyone. I'm so happy to be welcoming you to our, I think this is our fourth kidney-centric Discoveries in the Making. So the idea behind this is, like what Dr. Creel said, is to have a general audience to present some of the ideas that are happening here at UAB with some of our more advanced graduate students and postdoctoral fellows, and to actually provide that information to not only the students, the undergrad students, but also others in our kidney pipeline programs. So we have two kidney pipeline programs. One's called CURE, and those are our students that are undergrads from all over the United States that come here to UAB to do summer research projects, and our PROMOTE students who are pre-matriculating students into medical school, graduate school, whatever, pre-professional schools, and we're providing team science opportunities for them. And so in both of those NIH-funded programs, we provide this opportunity of Discoveries in the Making so that people can get a feel for what it's like to actually present science to a general audience. And that's the whole idea behind it. And to make sure that we get the word out about kidney disease and all of the cool research that these students and fellows are doing. So I thank you very much for coming. And you're in for a delight here to hear about these three research projects.
Thank you so much, Dr. Bunten. Mary, if you'd like to go ahead and start sharing your screen, that would be awesome. We have three fantastic presenters for you tonight. The way that we will order this is to have each one share their materials for about 12 or so minutes, and then we'll have around 5 to 10 minutes of questions. If you are not comfortable raising your hand or unmuting yourself to ask a question, please feel free to put it in the chat. We're more than happy to facilitate questions that pop up in the chat. So I'd like to introduce you to Mary Bunten, and I'm going to allow her to take it away.
All right, can everyone hear me? Awesome. All right. All right, here we go. So hi, everyone. My name is Mary Bunten. I'm a third-year PhD student in graduate biomedical sciences under the mentorship of Dr. Matthew Renfrow. And today I'm going to be discussing a kidney disease caused by mistaken identity, aka IgA nephropathy. And to give you a brief outline of where we're headed today, we're going to tell you why we care about the kidneys, how the kidney functions, just a brief basic overview, and what signs may lead you to think there are kidney filtering problems. And then what is IgA nephropathy and how does it work in the body, as well as what our group investigated to understand this disease even better. So we know why we should care, but why you should care about kidney disease. The NIH invests nearly $39.2 billion annually in medical research, with about $619 million of that being designated to kidney disease. However, in the US, kidney disease costs about $110 billion in taxes per year, with about 30 million Americans having chronic kidney disease. So 40% of our IgA nephropathy patients are in need of kidney dialysis or a kidney transplant. And in Japan, 1 out of every 2 kidney biopsies is this disease. This leaves our patients with a lower quality of life because there's no prediction of prognosis or how the disease is going to go on. And it leaves them with very high medical bills for treatments, medications, if they need dialysis or a kidney transplant. So it's really important that we study these kidneys and get answers. So a brief overview of how the kidney functions. I know we all know this, but the kidneys are 2 bean-shaped organs on either side of your spine. And within the kidney are lots of these little filtering units called glomerulus or glomeruli. And so they take in the blood from the body, they filter out all the waste and send it to the urine. And so throughout this presentation, we'll be kind of making it akin to this coffee filter and coffee pot. And so our old blood goes into the top where this filter is. The coffee filter in this case is our glomerulus. And this glomerulus makes sure that nothing gets through into the urine that shouldn't be there, like proteins or blood. And so it filters out all of that blood and then it sends the rest of it into the bladder, which is your waste. So we'll be focusing on the filter part of that for today's talk. So what are some signs that there could be kidney filtering problems? The first is that your urine could look cloudy or foamy, lots of bubbles in it. So here you see an example of clear urine, not discolored or anything. And then you come over here and you see this urine, it's kind of cloudy, hard to see through, has some bubbles in it. And so this is evidence of protein leaking into the urine that shouldn't be there. You should not have excessive protein just leaking through your filter into the urine. And once again, showing this coffee filter. Sometimes if a filter becomes overwhelmed, it gets a tear in it and then coffee grounds can seep through into your coffee. So that's the same thing. If the glomerulus gets a tear or gets broken down, it can leak proteins or blood into the urine. So that leads me to the next sign of filtering problems, which could be dark tea-colored urine or cola-colored urine. This could be evidence of blood leaking into your urine. And so one way they test for this is using a dipstick. And this can tell how much blood or protein is in the urine, if it meets normal levels or if it exceeds them and makes them think you might have some filtering issues. And so now we're going to jump into IgA nephropathy or IgAN, as I'll be referring to it today. It's the most common type of inflammation in the glomerulus, your filtering unit. And it can happen in both genders, but it's most common in males. And common onset of the disease, when you start seeing symptoms of it, occurs in the 20s and 30s, but it can strike at nearly any age. And a lot of times it doesn't cause symptoms in early stages, so it can go unnoticed for many years. And 40% of our IgAN patients need dialysis, so intervention of filtering of the kidney or a kidney transplant. And once again, the symptoms of this can be blood or protein in the urine. And so this is to kind of show how they do diagnosis, which is by a kidney biopsy. They take a piece of the kidney tissue, they kind of cut it in half basically, and you'll see a nice picture of the glomerulus like I just showed you. And so up here for your reference is a healthy glomerulus. You see all of these nice open circles, they're round, they look just really nice and open wide. You come down here into the injured glomerulus and we're not seeing those wide open crystal clear circles anymore. Some of the circles look kind of broken down or they're just not there at all. So either they've been crushed or they've been blocked up. So you're not filtering anymore freely through the blood vessels. So what happens in IgAN? So we call it a case of mistaken identity. IgAN, IgA nephropathy, is named for Immunoglobulin A, IgA. It's an important immune protein that helps fight off sickness. And in a normal IgA molecule, you'll see these little galactose sugars attached to it all over it. And then in IgA nephropathy, we don't see those sugars, or we see a lack of those sugars. So it's deficient in galactose. You may not see any of those sugars on these IgA molecules. And so now that it's missing that identity, the body isn't recognizing it as self anymore. It's seen more as like a foreign invader or something that shouldn't be there. And what does our body do when they sense a foreign invader? It attacks. And so this is where the mistaken identity comes in. This IgG, immunoglobulin G, just unintentionally recognizes that IgA, sees that it's lacking sugar, and thinks that it's the enemy, it's an invader. And so it goes and it attaches to it and forms an immune complex. So just two immune proteins coming together and complexing, and this forms in your blood. And it travels all the way into the glomerulus, where it can break down those nice open vessels like we looked at before. And so it breaks all of those down, causing a tear in your coffee filter, so that protein and blood will start leaking through that filter into your urine. So we understand that these two immune proteins are important to the disease, but what we don't understand is there are other blood proteins that associate with these complexes. So we know that they're there, we just don't know what they are or how they contribute to how damaging these complexes can be. And so we're trying to answer that question. What proteins are there and why do they cause such destruction in the kidneys? So we had a very simple experimental structure I'll walk you through right now. We took blood samples from patients and healthy controls, and then we sucked all the red blood cells out to get what we call serum. This serum is then sent through what we call size exclusion chromatography. And to put it simply, you have this nice long tube we call a column. Inside that column are a lot of tiny little beads, and the larger proteins, they don't get stuck in those beads straight through. But in the proteins, they kind of get stuck between all of those beads and columns, so they move a little slower. So we're able to separate out these large complexes, so we end up with a sample that only has the complexes left in it. And then we use mass spectrometry. So these immune complexes go into our instrument, they ionize, or we give them a charge, and then those charges are detected so we can detect what proteins are there in that sample. And so what were we able to learn? We learned that there are several other blood proteins, about 80, that we commonly see across our samples, but they primarily come from the complement cascade. And so this is a part of the immune system that enhances antibodies to kill or clear invading or foreign pathogens. And so what do we have in this disease? We have a foreign invader that our body isn't recognizing. And so the immune system is sort of hyping it up to go and find these sugar deficient IgA. And so these are complexing somehow with these immune proteins that we already knew, causing these damaging deposits. And so where we need to go from here is we need to figure out, we know what the proteins are. What happens if we get rid of a protein? Is that complex still going to damage the kidney? Is it still going to cause your filter to break down and leak protein and blood into the urine? And so we're going to look at that, what each protein's role is in how destructive these complexes are in the kidney. And so I'd really like to thank you for your attention today, and I'd be happy to take any questions.
Thank you so much, Mary. If you have a question for her, please feel free to unmute yourself and ask, or we can moderate and facilitate the questions in the chat. We have about 5 to 7 minutes, so happy to take those. Just to get you started, Mary, how did you get interested in this research and why are you passionate about it?
I'm passionate about it because I know how badly kidney disease really affects a lot of people, not only in our country, but in our communities and in our families. And it doesn't have a lot of answers right now. There's a huge amount of questions. And I think that, I have people personally in my life that have had kidney disease. So it really hits home when you have an opportunity to look at a disease that's so common in so many people's lives and say, I can try and get an answer for that. And especially with this project in particular, it's almost like a puzzle, trying to see what all the pieces are. And now I kind of have to work backwards. How do they all fit together? How do they all work? And so, yeah, it's a big mystery, but it's going to help further our knowledge of the disease. And I think it's really cool to be a part of that to help these people get more answers.
Thank you. I think Dr. Pollack has a question for you as well.
Oh, I think somebody else was before me, but I'll go quick. So Mary, I really, really great job. It's not easy to explain sometimes your research to a general audience. So it's very cool how you put that together. But the question I was going to say was we have a lot of students on the Zoom today that are potentially, could go into nephrology or kidney research. And so I was wondering if you could tell us in just a few sentences, kind of what I know that you're working on a fellowship. And I was hoping you could tell us a little bit about what the main point of your fellowship is going to be.
So the fellowship as in the project, or what I'm doing in the fellowship specifically?
Or just the project. So how this relates to the project that you proposed that, of course we want NIH to fund that project. So can you tell us just a little bit about that?
Yes, so the overall idea of my project is we're trying, since this is a broad spectrum of disease, it looks different in every single person that has it. We're trying to find, are there certain proteins in these complexes that fit a profile of like clinical characteristics or how the patient is progressing in their disease. So if it fits one and someone else has the same sort of characteristics, we can say, maybe that protein fits another, and we can sort of create what we call phenotype, or a picture of what this disease could look in certain patients. We all have learned through COVID how important it is to have screening for diseases. And this could be a way of screening people for this disease before it progresses to the point where they need a biopsy, which is very invasive. This is just a blood sample, so it's much simpler. So that's kind of where we're going with that.
Awesome, thank you. We have questions from Hannah Cleary, and yeah, we'll start with that one.
Oh, okay. Can you go back to, I think, your methods slide?
Yeah.
Okay, so I might have totally missed this, but like in your animation, how the larger proteins are like moving like farther than the smaller proteins. I might have just completely missed that, but I wanted to know why that was, because I feel like usually larger proteins like stay like closer to like the source, and then the smaller ones move forward.
That's a great question. So this animation is coming from this part of the instrument, which is this giant, what we call column. And that column is filled with thousands of little tiny beads. And so all of these gray dots represent all of those little beads inside. Smaller proteins are able to interact. These beads have like tiny pockets, and those smaller proteins can fit in those pockets and kind of hang out in the beads a lot longer. The larger proteins don't fit in those pockets, and they kind of just push through like a steam train through those beads. So they just come down really quickly and separate first. And so it's just a way of separating out everything by how big it is. Does that answer your question?
Yes, thank you.
Yeah, and I'm the next one with the question. My name is Zsuzsa, and Mary knows me. I'm her team director. So beautiful presentation, Mary, and I'm very happy to be here. And I really like your approach, and it's a very interesting thing. So as a classic pathologist, IgA nephropathy is a classic mesangial proliferative glomerulonephritis. So the mesangial cells are proliferating because all those immune complexes are deposited in the mesangium. So can you find the complement components there, and what is the difference between the patients? Is it is it a difference in the complement cascades? You know, there are different types of the classic and the alternative pathway of activation of complement. So which are there differences between the patients in that? And the question is, do you really find those in the mesangium as well?
Okay, so yes, there is a complement cascade. The alternative pathway is usually the one that presents in IgA nephropathy, and it's the one that we get the most proteins from. We don't see as many that are associated with like the classical. And so basically they have looked at mesangial deposits. So when those complexes go into the glomerulus, and they have seen in those biopsies, they have seen complement factors. So this is kind of good to know that the complement initiation doesn't just start in the kidney, but it starts in the circulation as the complex is forming. Does that answer your question?
Yes, yes. Congratulations. Good job.
Mary, you have a few questions in the chat. The first one is from Jared. So how does the immune complex form? Is it just two immunoglobulin isotopes stuck together?
Yes, so it's that IgG molecule binding and then binding to the hinge region of the IgA molecule. So it literally just attaches. And then the second one is, how does the accumulation of this immune complex damage the glomerulus? Great question. So if you think about it, if you have an accumulation of these big immune complexes in this small filter, it's going to block that filter, or it's going to start breaking that filter down. As well as Dr. Bebach was saying, when it does get in there, it has this effect where it can cause the mesangial cells, so the kidney cells, they just start replicating over and over and over and over again. And if you think about it, if you keep replicating, it's going to burst. It's not going to fit all of those cells in it anymore. So there are several ways that it damages the glomerulus. And then Marina says, is it possible that this disease is part of an undiagnosed autoimmune disorder that manifests itself as IgA nephropathy? So IgA nephropathy is considered an autoimmune disorder because you have the IgG finding those that aren't recognized as self. So it's its own autoimmune disorder. And I think that's all in the chat.
Great, thank you. Dr. Bebach, did you have one more question?
Oh, no, thank you.
Okay, great. Okay, if no one else has any more questions for Mary, thank you so much. And we will allow Leslie to start sharing her screen.
Thank you.
Awesome.
Hey.
Welcome, Leslie.
Thank you. So my name is Leslie Donoghue. My talk today is called Tissue Chips and Microphysiological Systems. A little bit of a mouthful, but I'll do my best to explain these technologies to everyone. And I'm a fourth-year PhD student in biomedical engineering in Dr. Palaniappan Sethu's lab. Quick outline. We'll go over the basics of tissue engineering, their applications, the definitions of tissue chips and microphysiological systems, their applications, and why they're advantageous within the biomedical sciences. And like I said earlier, I'm a biomedical engineer, and the field of BME, for short, is very broad, but the areas in which I have the most passion for is what is called tissue engineering, where the goal is really to create a functional human tissue from cells in a lab setting. Much of this work occurs in an in vitro environment where the cells are no longer in an organism and are grown elsewhere, such as this flask. When I first heard of the in vitro and how to distinguish it from the other Latin terminologies way back when, just think of IVF or in vitro fertilization, where that fertilization is occurring outside of the body. So although we are not trying to create babies within our tissue engineering, we are trying to mimic certain aspects of the human body. And to do this, we have to pay attention, pay special attention to four things. And the first and most obvious are our cells. We require the right cell type, and this is also special consideration if we just use one cell type or multiple cell types. And the body alone has over 100 different cell types. And they also, we have to take special attention of where we get our cells. So we can harvest these cells for in vitro from human tissue. We can commercially buy them from vendors, or we can also harvest these stem cells and reprogram them, which are essentially a precursor cell. So it's not one of those 100 plus specialized type of cells. It's really a precursor to that. So it can become a nerve cell, it can become a heart cell. And those there's a couple of different ways you can do that, but the technology is these stem cells. The second thing we pay special attention to is this extracellular matrix, or ECM. And ECM is basically the right environment for the cell. And like in the movie The Matrix, there's a lot going on within the matrix of any particular cell. But essentially the ECM, which is also called a scaffold, is there to support the cell in its existing environment. So this is the second consideration tissue engineers pay attention to. The third is the communication, or the Zoom call of the cells. So cells not only communicate with other cells, they also communicate with the ECM. And it's through these certain biomolecules that make cells in a healthy condition, or even a disease or pathological condition. The last but not least thing that we are taking special consideration of is of forces. And in the body, like in Star Wars, there are many forces all around us. And these are mechanical and physical forces that are there to influence the development of cells. And this is an area which my lab space probably studies the most, is what do these physical forces are doing to these cells. Specifically, the area of which we study is called microfluidics. It's just like the name suggests, micro as in, or small as in micro, and fluidic as in fluid flow. And our bodies have a lot of fluid moving at any point in time. So it's really important to model this dynamic behavior. Besides fluid flow, there's also a number of forces at play. So we do our best to model all of them in our devices. And this is where the really heavy engineering concepts come into play. With these four foundational concepts, biomedical engineers can get to creating tissues, but there are two broad areas of research, with the first being implantable tissue, with the goal of this actually going inside of a human. And here's an example of a tissue engineered bladder. It's one of the few organs that's already being used to supplement organs on the transplant list. So there are people walking around with these tissue engineered bladders. So this is kind of that first area that branches off for tissue engineering. But the second is being tissue chips in microphysiological systems, the area of which that Dr. Sethu Labs likes to create. And here's kind of the working definition. It's an in vitro device that can be used to model both structure and function of working units in the body. So here we combine the cells, the ECM, the biomaterials to enable communication, and and the forces at play, so that they make these small little chip devices specific to this tissue or that organ. And to better define the distinction between tissue chips and microphysiological system, MPS, or the shorthand for it, is really just a next level complexity of tissue chips. So you could have multiple tissues connected to study those interactions. And to use these technologies, they have the capability to screen drugs, model disease, and are part of this kind of personalized medicine movement. And specifically, I am focused on the cardiorenal application of these technologies. But first, I will describe tissue chips and MPSs in the context of drug screening. And the rationale is actually quite interesting. As it stands today, the current drug discovery and development system is very, very slow and very, very costly. It's also highly difficult to have an FDA-approved drug to go from start to finish, with only 1 out of 10,000 drugs being approved by the FDA. And for that single drug, it costs over $2.5 billion in between 10 and 15 years to get to market. So why is the success rate so low and the failure so high? Largely because humans and animals are very different. And the failure rate, if you have a successful animal model, it will still fail 90% of the time in the clinical setting. And a good example that my PI likes to give when he's giving talks is aspirin, which is very common in all of our households. And if it was, say it didn't exist and we were trying to go through the current FDA process, the levels at which it's effective in humans would be toxic to rats. So it wouldn't get past that animal model stage. And the reasons is because of toxicity. 30% is why it fails, and not specific to aspirin, but overall 30% of the failures of drugs is because of the toxicity. And then the 60% of the time is the failure of efficacy, or how well the drug is supposed to perform. But the point I'm really trying to make is, as a society, we're wasting a lot of time and resources with this existing workflow to create drugs. So this is kind of where, or organs on the chip, which is just another synonym for MPS or tissue chips. But to be clear, tissue chips and MPS systems are not there to replace animal models, but they're really to supplement and alleviate some of the economic and time burden of their current drug discovery process. And since they are made of human origin of the cells, hopefully we can better screen drugs than the current process. So it's almost like a parallel. And statistically speaking, many of the drugs in the hospital setting are toxic to the kidneys, otherwise known as nephrotoxics, with the off-target effects occurring in 20% of the patients with acute renal failure. So much of my work revolves around the proximal tubule of the kidney, which is that first section after the glomerulus that Mary was kind of talking about. So you have that coffee filter, and then the first section that filtrate goes through is called the proximal tubule. And it's an area that's really highly susceptible to drug toxicity. And so largely because there's a high level of concentration of toxins in that first area. So now you can see an example of one of our tissue chip systems, and it contains a bunch of elements I won't explain, but just know that we pay special attention to the cells, the ECM, the communication, the forces, and all the major design criteria to replicate this portion of the kidney. And then also specifically, you can see how flow, i.e. one of those physical forces, which here is labeled the dynamic environment, changes how those cells behave through a number of different markers compared to cells in a dish in the static condition. Secondly, our lab likes to model disease conditions with our tissue chips. And our lab has a cardiac tissue chip to study heart failure, specifically conditions with pressure and volume overload. And to put it simply, you have a heart that can experience too much pressure or too much stretch, and it will change how those cardiac cells are behaving and functioning. And here's an example of our cardiac tissue chips. It contains these small fibers that consist of the appropriate cardiac cells, appropriate ECM, the markers for communication, and the forces similar to seen in a real human heart of the pressure volume overload. And after subjecting these tissues to stimuli, we can conduct downstream analysis to see how closely we are actually mimic the human heart tissue within the human body. Third, I will very briefly discuss personalized medicine, which is the last application of these tissue chip and MPS systems. And essentially, it's you on a chip, and it's a very, very lofty and ambitious one, but tissue engineers have this long-term goal of having someone's own cells in an in vitro environment. And they would do this through that stem cell technology that I briefly mentioned earlier. So not only they would be your own cells, but they would also be from all different tissue and organ types connected together. And you ask, like, why would you want to do this? It's because they're trying to see if different treatments might be better for you, because we're all different, and one treatment for you might be harmful to another. So our lab is really doing some work within the stem cell engineering to do this personalized medicine. And one of the ways we're doing that is through the stem cells of functional heart tissue. It's something I've worked more so in the past and that my lab mates are doing currently, but here on the right, it's very brief, but you can see the slight pulsating, and that's basically the stem cells that are cardiac tissue beating on their own after 8 days. So this is one of the technologies for personalized medicine. So this wraps up my presentation. I would like to thank everyone in the funding on this slide, and hope everyone was able to learn a little bit more about tissue chips, NPS systems, what goes into developing, and the rationales behind them. And I would open up the floor for questions.
Thank you, Leslie.
Rahman?
Hi, Leslie.
Hey.
Really nice job. Very, very wonderful, but very difficult topic to try to give to a general audience, but you aced it there. So I do know that some of your own personal work is related to how you can try to model a kidney in some of these. So can you give us just a few, just a quick response on how that work is, a quick update, I guess, on how that work is going? Because I know that that's part of your NIH fellowship that you got funded. And I think people would be interested in that.
Sure. So it's kind of a model building on itself. So we kind of start with as few variables as we can at a time. So if we can get our proximal tubule epithelial cells in a environment where they're showing markers in this dynamic environment, and we have ways to screen how healthy these proximal tubule cells are functioning when we add drugs or add a disease condition. So the current model is to incorporate a vascular network, which is a limited area of research with these existing proximal tubule chips. So a lot of times they don't have the full functions of the normal proximal tubule. So we're trying to advance on those reabsorption, those secretion functions that are essential to be a true proximal tubule. The first question in the chat I see, is there a model of off-target effects in a chip? So I think the best way to do this is kind of this NPS system. So you could, it really gets complex because you could have your liver, you have your kidney, you have your heart. And so like if you have a drug that's metabolized by a liver and it goes to the heart, does it have that same cardiotoxicity? So putting those chips together, so there's people in our lab working on different tissue types and organs. That's the best way to model our off-target effects as of right now, but it's definitely in its infancy stages. Hopefully that answers your question. What are some of the chemicals you use to cell scaffold in matrix? So that's a good question. A lot of the matrices, they can either be naturally derived, so you could have something that is collagen 1 from a rat tail, and that is in like a little bit of a tube, and you could mix it with the cells and you can get a gel. There's also some of these more synthetic, like a polyethylene glycol, but the negative of those is those aren't really natural to the human body. The one that I specifically work on is called gelatin methacrylate. It's kind of a hybrid because it's derived from gelatin, but it's also a polymer. But we can fine tune this gel to basically have the same kind of Young's modulus or the mechanical forces of a proximal tubule. It allows us to form these like natural capillary networks that I'm trying to incorporate into my dissertation. So there's a ton of different scaffolds and matrix. It's just, I've kind of divided those 4 areas to make a chip, and researchers are just studying those particular areas one at a time as well. So there's a whole field out there of different matrices. Okay, and then mention forces, but is there any method to determine ideal chemical messaging density? I don't know if someone can elaborate on what they mean.
Sorry, that was my one. I guess what I'm trying to say is I know that there are chemical messengers going between cells in within one particular tissue, but then also throughout the body. And on a chip, I could see where getting the density right would be important. And I was wondering, how does one determine the right density, and then how do you replicate it?
Yeah, the best example I have on top of my hand is like permeability studies, where see how like these chemicals make a gradient, and one way you could do that is a fluorescently labeled dextrin. You can look at this dextrin under a microscope and see how well it's like perfusing within a tissue type. So and how that relates to other tissues as well. I'm not sure if you've heard of fluorescently labeled dextrin, but you can basically change the kilodots in size of the dextrin to mimic the protein you're specific in, or that at that size or whatnot. So that's one way.
Hi, Leslie. I raised my hand, but I'll just jump in there. That was really cool. I mean, I'm just impressed by people to take on such a challenging effort. But I was just kind of commenting to make a comment about the last question. I know that drug companies all screen a lot of their drug candidates, well, really early on in cell-based assays, and they can measure things that those cells make. It seems to me that if you could model a better, something a little more complex than just a cell growing on a plastic plate, that would be really great. I don't know, have you heard of any companies doing any kind of thing like that, using these model systems for that sort of work?
Yeah, it's again because the process is so long that the drugs that you are kind of hearing about now in the clinical phases are, I mean, they did all those in vitro studies way back when, but it's definitely kind of, companies are cognitive of these technologies, and some of them are commercially available. So if they could use those to potentially screen them better on. The simplest nowadays, it's like kind of a loose definition of a chip, but people use transwells all the time just to mimic a couple different cell types at once. But I think this research is really only about less than 15 years old. So there's definitely a way to go. So
Great, thanks.
Okay, awesome. I think we are about out of time for questions for Leslie. Thank you so much. Next up, we have postdoc Paramita Pati, and we will ask her to go ahead and share her screen and give us her presentation.
Hi, everyone. So I'm going to talk about how the timing of when we eat and what we're eating how it can affect our cardiovascular health this evening. First, I'll talk a little bit about circadian rhythms. So what are circadian rhythms? Usually we think of when we wake up and go to sleep, or the sleep-wake cycle, which is the most well-known circadian rhythm. So all organisms on Earth, ranging from bacteria to plants to mammals, including us, have all developed this 24-hour system in which we are tuned to adapt to the rising of the sun and the setting of the sun. So with approximately 12 hours of light and darkness. And there's actually a molecular mechanism throughout the cells in our body, in different tissues and in the brain that helps control these rhythms. These rhythms are important for many aspects of physiology in the body. So from body temperature to the time at which we're most coordinated, to when we have melatonin secreted, which helps us go to sleep at night, all of these change based on the time of the day. Blood pressure rhythm also has a clear day-night difference. So we can see here on this graph on the right that systolic blood pressure and diastolic blood pressure in people is higher in the daytime or active period and lower during the nighttime or inactive period, the time when most people are sleeping. And there actually is a circadian pattern of bad cardiovascular events, including strokes and heart attacks, which are actually most frequent in the early morning. So there are time of day differences in blood pressure. And these time of day differences are important for physiology that we maintain when we're eating. A number of studies have shown that night shift workers have a higher risk of cardiometabolic disease. And the loss of circadian rhythms, it leads to disruption in metabolism as well as in cardiovascular rhythms. So eating at the right time of day is important for our health. So as human beings are diurnal, right, the active phase is in the daytime for us. But when we have a shift in food intake or meal timing to the late night or the normally inactive phase, we have disruption of our rhythms. So changing the time when we eat to what goes with our body clock can help our cardiovascular system and overall health. Recently in humans, there have been studies that show that time-restricted feeding does indeed provide cardiovascular events. So these are people who are, subjected to only having their meals available through, before the evening, basically. And we see that systolic blood pressure is decreased, as well as diastolic blood pressure, and 8-isoprostane. So this is a marker of oxidative stress. So oxidative stress, it leads to negative changes in cells, and it can affect metabolism in many bad ways. So we see that in human beings, that this meal timing, it indeed doesn't have these good effects. So also, not only when we eat, but what we eat is also important, right? So obesity is a major risk factor for the development of cardiovascular disease. And this includes an increase in oxidative stress, as well as blood pressure, as well as cardiac hypertrophy, right? So how the heart structure is changing. And it can also even increase vascular disease and how well blood vessels function. We see here a diagram of a blood vessel, and we see that there are many different parts in the tissue, right? There are these different structures, including the endothelium, which is a thin layer of cells that is very important for how well blood vessels can relax, as well as the smooth muscle and these different structures. So there can be changes in all of these different structures in disease conditions that can lead to worsened outcomes. We see also that, different factors, right, including diet and obesity, as what they can lead to increases in systolic blood pressure, and this can lead to increased stiffness in the big artery or the aorta. And these can in turn also lead to cardiovascular disease. So this will be important later as one of the measures we looked at in our study. Our overall research question is does the timing of food intake during a high-fat diet improve cardiovascular health? And we hypothesized that restricting the timing of food intake to the active period will improve blood pressure rhythms and vascular disease in mice who are eating a chronic high-fat diet. I will give you an overview of our dietary protocol. So these are male 57 black mice. So this is a very common type of mouse model used for physiological studies. So they were either given a normal diet or a high-fat diet for 20 weeks or for 5 months. And then at the end of this time, they were given food only during the nighttime. So I should mention that mice and other rodents are nocturnal. So their schedules are the opposite of most people, right? So they're sleeping in the daytime and more active at night. And so they only had food available at night. So they had no food in the daytime between ZT0 and ZT12. So ZT means Zeitgeber time. So this means when this is relative to when the lights come on. And then, so the lights go on at 7 a.m. and they go off at 7 p.m. And this will be important for some of the other measures we looked at. So we first looked at the blood pressure rhythm in these different groups of mice. So similar to in people, we can measure blood pressure with a catheter put into an artery of a mouse, but we can actually measure this continuously by a technique called radiotelemetry. So this catheter, it's implanted into the mice and their blood pressure is measured every 10 minutes for many days. So it can be measured all the time, continuously. And we can see here that in the mice on a normal diet, their blood pressure is lower during the time when the lights are on, and then it is higher when the lights are off, which is the gray period or the night period. So they have a clear day-night difference in their mean arterial pressure. We then looked at mice that were on a high-fat diet, and we can see that their blood pressure does not go down as much in the light time, but that when we give them restricted feeding, their blood pressure in the orange line, it begins to dip more during the inactive period. So time-restricted feeding, it helps improve the blood pressure rhythm during a high-fat diet. I mentioned aortic stiffness earlier. So this is a marker of vascular disease, and we can measure this with the technique in the mice during the study. So we saw that in the mice on a high-fat diet, the vascular stiffness was much higher. So these mice are at greater risk of developing cardiovascular disease because they have more stiffness in their blood vessels. We then wanted to actually look at the structure of the blood vessels because we saw these differences with the arterial stiffness being higher. And so these are histological stains. So we use 2 different kinds of staining to do our analysis. And we can see here that we have the normal diet group. And then once we look at the high-fat diet group, that the arterial wall of the aorta is much thicker. So I mentioned earlier that blood vessels can undergo different changes in the structure, right? In the different types of cells and tissues there. So this is a type of arterial remodeling. And we can see that the high-fat diet, it increases the thickness. And then we also saw that the wall thickness, it's lower with the time-restricted feeding. So after only 2 weeks of time-restricted feeding, after this 5 months of high-fat diet, the aortic walls look more normal in the mice under the time-restricted feeding. And then I mentioned we'd use 2 types of staining. We can see here in this blue stains that with the high-fat diet, there's more pink compared to the normal group, as well as to the group with time-restricted feeding. This group looks more blue. So there were other kinds of differences in the types of, molecules and cells in these mice after the restricted feeding. I mentioned oxidative stress earlier. So in humans, it was shown that oxidative stress can be lowered by time-restricted feeding. We also found that this marker, 8-isoprostane, is lower with time-restricted feeding in mice in our study. So time-restricted feeding, it clearly has beneficial effects, as we've seen in a mouse model of obesity. So to conclude, if the mice don't eat during their inactive time, the time they should normally be sleeping, we can help reverse some of the negative effects in the blood vessels, as well as decrease oxidative stress. And similarly, in people, these findings are, have been recently shown, and further work needs to be done. And we are particularly interested in looking at some of the biochemical changes and pathways involved to these changes in physiology and these different markers during obesity. So thank you, and I would appreciate any questions.
Thank you so much, Dr. Pati. People can raise their hands if they have questions, or you can jump in, or you can put them in the chat. Rita, I know that if you're working on this as your postdoc project, it's probably been a long road for you. And what are some of the things that got you interested in this project, and where do you plan to take it?
Yeah, so this project has been a large part of my postdoctoral training, and it's actually the major project that I was working on. And today I highlighted some of the major findings. So I think there's a lot more work to be done. Looking at the mechanisms that lead to these changes in physiology that we observed. And we've started some of that work, which I didn't focus on today, but there's definitely a lot more to be looked at the different signaling and the time of day changes and how these all kind of relate together in the body between these different organs. We've also, we've focused in the kidney, we've focused in smaller vessels. So there's quite a lot, as well as more functional studies using the aorta.
Awesome.
So if I could chime in here real quick, I don't know if you want to relate this to the current idea of intermittent fasting and how this could be a model for that, or think about kind of if you could relate that to this.
Yeah, so I think this our findings, right? They have a lot of relevance to a lot of these different ideas about different diets, right? So this idea of intermittent fasting. So there are different kinds even, right? There are some that people only eat certain times in the day, and then also where there are alternate days of fasting. So I think this is something very relatable to a lot of, a lot of things that, you know, people might do in their daily lives.
And how it might relate to kidney disease, right?
Yes, so we also are, that's something that we're also looking at, and we see that it you know, it definitely, there are differences with improvements in kidney damage.
You also have a couple of questions in the chat.
Let me see what the questions in the chat. Yes, so one question. Yes, so yeah, we definitely, you know, we're looking at some things with Dr. Young. Okay, what time of day would you be most interested in studying further in the future? This is a great question, actually. So we, the studies I've shown today, so the blood pressure, right? That's something that we looked at over a whole day, right? Or multiple days. And then when we looked at these other changes, right? Those were at a single time point. We also have other data, which I did not focus on today, but it was looking at how well blood vessels relax or constrict. So whether during the inactive period or the active period. And we found that time-restricted feeding improved the blood vessels function during the inactive period. And so given that finding and the data I showed today with the isoprostane being lower during the inactive period, I think that a lot of the changes in this particular model we're seeing are in the inactive period. So the middle of the inactive period. I think that that's where we would focus for future studies with this particular model. If that answers your question.
Awesome. You had a couple more. Do you think people who live in areas that don't receive as much sunlight or more during some seasons would be affected?
I think, yeah, I think so, right? Because I think it depends, right, what their own schedules are really. So, you know, there are some places, right, in the more northern parts of the hemisphere where there may be very few daylight hours, right, and a lot more darkness. So I think that it would depend, right, when they're going to sleep, when they're waking up, or similarly, like places where there's a lot of daylight and people like to stay up all night, right? I mean, I think this is a great question, and I don't think anyone's really looked at these, you know, these kinds of readouts with in people with different lengths of day. But there are studies I should mention in rodents looking at, you know, different longer periods of light or shorter periods of light or alternating periods of light. And definitely there are metabolic differences. A question, have sex differences been looked at related to these findings? So that's not something we have done so far with these particular readouts, but it would definitely be good to do in the future. What do these and similar studies mean for all the people who are studying rodents, nocturnal animals in their studies? Well, I guess that people who are doing these studies also have their circadian rhythms disrupted. Speaking from experience. But I think, you know, again, I think short term versus long term, right? I think that's kind of the key here.
Yeah, I was actually thinking about what Leslie said earlier about how so many preclinical studies don't predict what's happening in humans. And maybe it's because when we study the nocturnal animals, we're not studying them at the same time we're studying the humans in terms of their 24-hour day. I just think it's something interesting to look at.
Yeah, no, definitely. Absolutely.
Awesome. Thank you so much. I don't know if you want to say anything else, Dr. Pollack, to round us out and thank everyone for coming.
Happy to. I really appreciate everyone sticking around and all your questions, very insightful. It's really always exciting to be a part of any kind of science learning. And I think this is a great venue and for discoveries in the making for us to all understand any level of science is always wonderful. So I appreciate everybody's time, and I'll see you around the labs.
Awesome. Thank you so much to Mary and Leslie and Paramita. Great job, everyone.
Bye-bye.